It is our great pleasure to organize a two-day workshop as a part of a joint program of TechIP 3 twinning activity, a workshop and hands-on training on bioinformatics and its recent application, arranged by the Department of Bioinformatics, MAKAUT, West Bengal. This workshop is particularly intended not only for the students pursuing their postgraduate studies in bioinformatics or biotechnology, but even for active researchers pursuing PhD in the domain of structural biology, biochemistry, and even several multidisciplinary domains, and the faculties who are freshly planning to teach their students various topics on sequence analysis, protein-protein interaction, so on and so forward. So I am Dr. Unshaman Bakshi, Associate Professor of the Department of Bioinformatics under the School of Biological Sciences of this university. Is extremely glad to coordinate this two-day workshop as the moderator. The idea of conducting this workshop is to focus on the importance of databases that are used to store and organize biological data. Also, another important aspect that will be covered in this workshop in sequence analysis, which is process of subjecting DNA, RNA, protein sequences using more analytical methods, and to understand their features, functions, structure, or evolution. Our Honorable Vice Chancellor, Sir, Professor Soikot Moitro, is not present in the program.
Join করেছে.
Okay.
Has joined.
Okay. Okay. We are extremely honored. Good morning, VC Sir. Good morning, our Honorable Vice Chancellor, Sir. We are extremely honored to have with us our Honorable Vice Chancellor, Sir, Professor Soikot Moitro, gracing the inaugural program of this workshop. Sir, please convey your valuable message to enrich us about this workshop. Sir, please.
Hello.
So please wait, it will take few seconds to properly join. So there is some network delay. Yes, sir.
Okay.
So he has joined, I saw.
Hello, Prem Kumar.
Yes, sir.
Sir is joined.
Sir, there is some network delay and sir is trying to join. We should wait for 1-2 minutes, sir.
Okay.
Okay, Shomen, you may continue once he has joined you.
Okay. Before we proceed, it is our honor to welcome Professor Rajabanerjee, Director of School of Biological Sciences, to address the audience and illuminate us with his views on this particular workshop. Sir, please, sir.
So, a very good morning to you all, the participants. This is a great pleasure for me to jointly organize as a coordinator also with Dr. Unshaman Bakshi, who is the Associate Professor of Department of Bioinformatics. And actually, I am very glad to see one of my young friends, Dr. Professor Unshaman Bakshi, who is very close to our university, associate professor of Kolani University, and who is also an alumni of Bose Institute, from where we did our PhD. And this is basically to tell you, actually, it is nothing but a new thing in Department of Bioinformatics. It is we are organizing every year several workshops, hands-on, national level, under the uh, supervision or whatever I say, under the help support of the DBT, Department of Biotechnology, Government of India, and this time we are organizing this with TechIP. And today's perspective, when in the corona pandemic, the situation is tough, and it is really not to use all the time to go for the lab, wet lab. So we have to think in a different way. And the thing is, basically, what already Dr. Showman told us, we are living because the cell are functioning. And as the cell are functioning, that depends on the informations are stored in the DNA, and the DNA is transcript this to RNA, and which is translated to protein, who are basically the key player of the whole system. So in this particular case, what I have to say, I'm just showing one of the slides which I prepared for this. This is basically, I am working on the structural biology, and this is basically working on the protein structure, or the protein structure functionality. So what I feel is basically, as the protein is the main part participating the key role, where the DNA is basically considering or containing the main information. So we have to know little bit of this. But it is very difficult how we have to go for the protein structure. Why I am using this word protein structure? Basically, if you see our hands, we can fold in one direction, not in a different way. Why? Because the structure of the fingers and the clamping in such a way it is in unidirectional movement. So similarly, when a particular structure is doing some function, or when in a retrospective way, when a particular function is done, we have to wait for the structure. There are various terms we are hearing that is called the misfolding, protein folding, and this is a Nobel Prize award. And in the last word of the Nobel lecture, Anfinsen Sibi in 1973 uses this word with a very high emphasis. The amino acids, or the functions of a particular proteins are embedded within its structure, and the structures are embedded within the sequence. So it is very difficult if we do not understand the particular sequence, or which is called the primary sequence, or whatever the things. So if we go for the slide show, we can see this protein structure prediction. What we are doing is basically, nowadays we have to know the protein sequences, and from this is coming from the DNA sequences, and then some methodologies we have to use, on based on which we have to do the structure predictions. And actually, I think Unshuman will tell you more elaborately, but it is our all, we all know this thing, the central dogma. Central dogma, and after the central dogma, in the translation, we got this primary, secondary, tertiary structures. And this is the protein architecture, is basically, we have to see these are the very important things, are peptide bonds, amino acids contains there are carbon amino group, carboxyl group, and there are the different side chains, which is basically they play the key role. During the time when Anfinsen Sibi is working with this, there is another guy called Chow and Fashman. They said that this is the particular amino acids has particular preference for particular structure. So this is the best important thing that I have already mentioned. The Anfinsen Sibi's experiments in 1957 demonstrate that the proteins can fold spontaneously. And this is, if you have 100 residues are there, 100 residues, if there are n number of conformations can be present. If you know what is the definition of a conformation, conformation is nothing but it is the momentary arrangement around the CC single bond or CN single bond, whatever the thing under the physiological condition, there's the possibilities are 100 to the power n structure. But within a few seconds, even but that is a fraction of a second, within a nanosecond, even time scale, the protein out of the all boxes take only one particular structure. And the question is why? This is the main key of the biological researchers or the research people who are working with the biologists to understand these things. So there are different levels of expression. Already I have told you primary, secondary, tertiary structures. And this is basically showing in a short way the alpha helices and looks like this is a spiral staircase, the beta strands. This is something which is using in the DSSP form. And then what we have to see that there is a conformational flexibility. What I have told you, there is a basically the three dihedral angle we have to know phi, psi, and omega. And basically this phi, psi, and omega, if you see the structures, you will find that it is repeating and you will get the structure. Now we have to come to the very, very important. The plot is the Ramchandran plot and the Indian scientists who should get the Nobel Prize, and based on whose this particular plot or the graph, the whole structural biology is standing. So we are honored to have a GN Ramchandran, and this is showing that out of these four boxes, only this is the very useful region for the helices. This is the strands and other things. And all other, they are conformationally non-preferable or disallowed. So when we have to go for this thing, what are the techniques we can use? There are techniques. The techniques are experimental techniques. These are the X-ray crystallography and NMR. But these are very costly and time consuming and other things are there. So what can do? Once you have to know the particular uh, proteins, what you have, if you know the particular sequences, then it is very important is to structure prediction using the computational program. So that will tell you why that is important, because this is the fill in the gaps of the sequence and structure that is helpful in the protein engineering. And the most important thing is the rational drug design. So when we have to go for this, there are two methods for the structure prediction. One is the computer simulation based on the energy calculation, and the second one is the knowledge based one. So I just tell you within 1 or 2 minutes. That is the I hope the sequence after sequence analysis. This is one thing you should know, and I hope Unshaman will let you know. This is the knowledge based energy, knowledge based approaches. There's a homology, there is a modeling, there is a threading, and there are other methods are there. So what we have to do, first one is the called the energy minimization. If we take something, it then like you just consider the examples of the apple falling from the tree, and neutrons actually discovered the uh, gravitation, law of gravitation. So there are various methods are there, and there's also very limitations are there. So we have to go for the knowledge based, and this knowledge based is basically homology modeling. I have told you. So in case of the homology modeling, we have to know the homologous proteins. I think Angshuman will tell you. We have to know the backbone modeling. We have to go for the side chain modeling, etc. And this is usually the simplest method, reliable method. But the thing is, you must have to have a device. Just like if you go for making an idol, what the thing, they have a device, they put it, and they are just taking it out. But the one more important thing is, has been observed that even the protein has 25% sequence similarity should be there. So this is the other things. I'm not going into details. There are threadings are there when you do not have any particular model, when you do not have any particular device or template. Basically, I should use the word. You have to go for the threading techniques. And there are another one. This is very important. Once you form the structure, you have to go for its flexibility, conformational flexibility, how it is. So you have to go for the molecular dynamics is giving you only one thing. The difference between the X-ray and NMR is, in case of X-ray, you have to go for the free structure, but in case of NMR, you have to go for the solute solvent insert structure. So that is very important. And if as a student of bioinformatics, you have to know this thing. And the whole thing is depending on the Newton's second law. So a person who are working with the physics, who are working with the chemistry, who are working with the mathematics, all are welcome now to understand, to expose or explore the biological problem. So with this, I just want to thank you all, just giving a glimpse, and to Dr. Poti and Dr. Bakshi to continue the session. But my point is to tell you this thing is nothing but to have to think about only the wet lab experiment is not the sufficient, because it is not possible to go for NMR or go for X-ray for all the things, but you have to know the sequence first. And once you know the sequence, most of the informations are there. And now you have to know from Unshaman how you have to do the sequence analysis. This is a very critical part of the whole story. Thank you, Chauvin.
Thank you. Thank you very much, sir, for covering the major aspect of the subject and the idea behind to organize this kind of workshop for the student and researchers. Now, next, I welcome today our respected resource person, Dr. Unshaman Bakshi, Associate Professor from University of Kollani, to address the audience and briefly explain the topics that he will be covering in this today workshop. But before that, I would like to introduce Dr. Bakshi to you all. Dr. Bakshi is basically a biochemist, then turned into bioinformatician and a computational biologist. He obtained his PhD degree from Bose Institute, Kolkata, and thereafter has several postdoctoral research experience from abroad. Few of them, namely Bach Institute for Age Research, California, Indiana University, Purdue University, Indianapolis, and also a visiting research scientist in the Department of Computer Science, Baptist University, Hong Kong. He is the recipient of the Young Scientist Award from University of Kollani in 2014, and he has published more or less 100 research papers in reputed international journals and conferences. Currently, Dr. Unshaman Bakshi is working as an associate professor in the Department of Biochemistry and Biophysics, University of Kollani, Nadia. So, with this, I now welcome Dr. Bakshi and request him to say a few words on the training that you will impart through this today's workshop. So, Dr. Bakshi, please.
Thank you, Dr. Poti, for the nice introduction. And I would also like to thank Professor Rajabanerjee for introducing me. Professor Banerjee is my senior in Bose Institute, from where both of us did our PhD. And it reminds me of my long interactions with Professor Banerjee in Bose Institute. And so we had lots of interactions on different aspects of biology and bioinformatics. And also Professor Banerjee used to teach chemistry to all of us. So it was also a very enriching and fulfilling experience for all of us. So today I'll be talking about the basics of bioinformatics. First of all, am I audible?
Yes,
am I audible?
Yes, yes.
Okay, so I'll be talking about bioinformatics, the basics of bioinformatics. And so the bioinformatics, what is it? Many of you who are directly working in the field of bioinformatics, or who are having their masters in bioinformatics, or who are directly linked to bioinformatics, know that it is an interdisciplinary science. It involves the knowledge of mathematics, statistics, physics, chemistry, and absolutely biology. That means, in other words, we solve biological problems with the help of computers. Now, there is a notion that bioinformatics is a pseudoscience. I heard this from many people. It is not true. Bioinformatics is a standalone subject, a standalone science, which provides supports to all other disciplines. Bioinformatics provides supports to physics, to chemistry, to mathematicians, to statisticians, to computer scientists, everybody. Like a few minutes back, Professor Banerjee said that it is always very difficult to have the structures by X-ray crystallography or NMR. This is absolutely true. These techniques, which we call the wet lab biology or wet lab techniques, these techniques, they are very expensive as well as very time consuming. And also the rates of failures are much higher. On the other hand, bioinformatics, though it is a probabilistic science, it is a predictive science, it gives you an idea about the possibilities that you have in your hand. Say you have the amino acid sequence of a protein. Now, what you can do with it? You can extract many.
Sir,
Yes.
Sir, are you showing some PPT, sir?
Yes.
So it is not visible, sir.
I'm not visible.
Sir, PPT, sir, PPT presentation, sir.
No, I haven't started the presentation. It's just an introduction. I will start the intro. That means my presentation in a few minutes.
Yeah.
I'm just giving you a brief idea about the bioinformatics, the notions behind bioinformatics.
Yes, yes.
Yeah. So I'll be starting my presentation within a few minutes. So what I was talking about is that bioinformatics provides the support to wet lab biologists. Also, bioinformatics streamlines the long experimental or wet lab. I should not use the term experimental because bioinformatics is also an experimental subject, but the wet lab, long wet lab processes. So with these few words, and in this time of pandemic, it is really difficult to go to the lab, wet lab, and do experiments or wet lab experiments. Rather, many of the scientists are now relying on bioinformatics, which provides you many information. If you go to PubMed, you will find within the like the last 2-3 months, there are thousands of papers on bioinformatics. So that way you will be able to identify the role, the functionality of bioinformatics in modern science. So with these few words, I'm moving on to my presentation. I'm going to share my screen with all of you.
Sir, before going to share, we just complete our session, inaugural session.
Okay. So shall I wait for some time?
Okay.
Okay.
Thank you, Dr. for your concise description of the hands-on training that we will conduct in the next 2 days. So with this, we can end of the inaugural session for today. Now we'll go for the vote of thanks. I very much thankful to Honorable Vice Chancellor Sir, Professor Shoykot Moitro, for his inspiration, guidance, and constant encouragement to organize this kind of workshop. Thank to our director, Professor Rajabanerjee, for good support to this activity. I thankful to Dr. Sarojit Das, Head of the Department of Bioinformatics, for being a good support to this workshop. Very much thankful to Dr. Joya Bandyopadhyay, Head of the Department of Biotechnology, for being a continuous mental and organizing support to conduct this event. Thankful to my student, specifically Shoham Banerjee, my research scholar, staff, and my colleagues, who all helped me to make this event a successful one. And last but not the least, I would like to thankful TechIP 3 for their financial support and the entire webinar team, especially Mr. Prem Kumar, for his continuous support on this online platform. Now, I will request to Dr. Bakshi to proceed with the main segment of this workshop. And before going that, I also request to the audience to ready the computer or laptop and require software for hands-on session. And if you have any question, then you may write in question answer box, not in the chat box. So Dr. Bakshi, you may now start for your session formally. All the very best from the entire team of the MAKAUT for a great session onward. Thank you.
Thank you, Dr. Poti. I'm going to share my screen with all of you now. Is the screen visible to all of you?
Yes.
Yes.
Okay. So probably due to some technical issues, I don't have the full screen mode operative right now. So I think it will come. So I'm going to start.
Sir,
Yes.
you can see the option at the top, slideshow.
Yeah, I
menu bar.
That. Yeah, I did that, but it's not like working.
The slideshow, click, sir.
Yeah, I did that. I know.
Yes, sir. Okay, sir. Please continue, sir.
Yeah. So let me continue, and I think it will come.
So you can check by resharing, sir. It will be opened in the next application.
Sorry, I did not
Sir,
get your
it
point.
may be appearing in the next application. You reshare, sir. Kindly reshare it, sir.
Sorry, your voice is breaking.
Sir, close this screen sharing and reshare.
Okay. So let me let me start.
Yes.
It's not moving anywhere. So I don't know what is happening.
Sir, kindly open the Zoom platform and sir, kindly reshare it, sir. Share screen once again, sir.
Yeah, that is what I'm going to do, but it's not doing anything.
Unshaman,
Huh.
তুমি ওই স্লাইড শোতে গিয়ে স্লাইড শো বলছি স্লাইড শেয়ারটা গিয়ে যদি আর অসুবিধা হয় তুমি প্রেম কুমারকে পাঠিয়ে দাও না, ও তাহলে শেয়ার করে দেবে। সেটা কি তোমার পক্ষে সম্ভব? তুমি যদি ওকে মেল করে দাও প্রেম কুমার।
Yes, sir. Sir will be joining soon, sir, because he has some network delay. Network
আমি
issue.
তা বলছি, আমি I am not saying this thing.
Yes.
Just I think network connectivity is low.
Yes, sir.
That's why
Yes, sir.
the
Yes, sir.
huge
Yes, sir.
file is not to be uploaded. So my suggestion is, if he share, he can. Obviously, it is his personal property, IPR Act. So if he can share in the PDF form, can you upload that thing or not?
Sir, let sir join once again, sir. Then I will ask him, sir.
সৌমেন তুমি সৌমেন
হ্যাঁ
তুমি একবার
হ্যাঁ
অংশুমানকে একটু ফোন করে জিজ্ঞেস করো যে ও এটা যদি প্রেম কুমারকে পাঠিয়ে দিতে পারে, প্রেম কুমার তাহলে আপলোড করে দেবে, এবং প্রেম কুমারটাও
হ্যাঁ
তো
সেটা করতে
আছে।
পারে, কিন্তু আরেকবার উনি
না
আরেকবার
না,
জয়েন
আমি
করুক, তারপরে দেখছি।
আই আন্ডারস্ট্যান্ড এভরিথিং, আই আন্ডারস্ট্যান্ড
হুম
এভরিথিং।
হুম
এটা তো অবভিয়াসলি লজিক্যাল একটা। হ্যাঁ ঠিক আছে তো।
Is it coming now?
ইয়া ইয়া ইয়া,
ইয়া
তুমি স্লাইড শোতে চলে যাও।
Hmm. Yes.
Yes.
So, okay. So now that I'll be talking first about the biological databases. So what is so important about databases, and what are databases? Databases are nothing but these are some structured collection of information. And the databases consist of certain units, certain basic forms of data which are predefined. The databases may contain information pertaining to several records, like, for example, in case of bioinformatics, like the name of the protein, like the length of the protein, the amino acid sequence of the protein, like the gene from which it is coming, and so on and so forth. So a perfect database should be very easy to search. For example, a database should not be like that. You have to have a dictionary to read a database. It has to be user-friendly. There has to be certain things which are called annotations. The annotations are nothing but the descriptions of the different types of data. And there should be the cross references. For example, if you have a protein in a database, and the details of the protein should be there in the database. Now, the experimental methods by which the protein or the sequence or its structure, whatever information are there in the database, the experimental methods are not stored in the database. So there has to be some other database to which this particular database should be linked. This is called cross-reference, so that you can have a comprehensive idea about the entire process. And the minimum redundancy. Redundancy means repetition. A database should not be like that the same kinds of data are present in the different databases. But in bioinformatics, this is one of the problems, the redundancy. We'll come to that. So what is the importance of a biological data? The importance is the popularity of bioinformatics started in the early 2000s with the advancement of sequencing technologies. More and more of biological data are constantly coming up, and there has to be some means of accessing those data. This necessitates the generation of biological databases. So what are the different kinds of biological data? There are the nucleic acid sequence data. That means the data pertaining to RNA, DNA, these nucleic acids. The data that are related to proteins, protein expression, their functions, structures, and so on and so forth. This is the central dogma of molecular biology. The genome, the DNA, is transcribed to RNA, which are then the RNAs are then transcribed, translated to proteins. And the proteins are the central players of most of the biological processes. So the central dogma is so important, and the concept of bioinformatics is heavily dependent on the central dogma. So the needs, why we need biological databases. This is the most important one, is to solve biological problems with the help of computers. In order to solve a biological problem, we have to have the data, the biological data. And as I told you in my previous slide, that the biological data are nothing but the sequences or the structures. In order to use the sequences or the structures with the help of computers, those information must be stored somewhere. The tools, the bioinformatics tools, access the data from the different biological databases. So this is the most vital aspect of having a biological database. Also, the biological databases are important to disseminate your own ideas, your own findings to other labs. This is the types or the different types of biological data. Mostly we are interested in this: nucleotide sequence, protein sequence, structural patterns, and the 3D structures. These are a kind of subsidiary to all these. These are the primary information, and these are the secondary information. Okay, by the way, if any of the participants has any question, you can just ask me. It's not a problem. You can type it in the question answer box. So the classification, we need to know what are the primary and what are the secondary or the derived databases. The primary databases takes the experimental results directly. The primary databases takes directly the experimental results, and the secondary databases take the information from the primary databases, and it generates some more data from the data that are present in the primary databases. So the basic difference is, say, let us take an example. Say from your lab, you identified the amino acid sequence of a protein. Amino acid sequence of a protein. Now, what will you do with it? In order to publish your paper, you need to deposit the amino acid sequence to a database. That database is the primary database. Now, once you deposit your data, your amino acid sequence to a primary database, what the secondary databases do is that they extract certain features from the primary database and they store all those features in the form of annotations in the secondary databases. So there is another way to classify a biological databases. There are certain biological databases which are publicly available and there are no restrictions. That means you can easily extract the data from there. There are certain other biological databases where you have the data, but you can see the data but cannot download it. So there are certain copyright issues associated with all these databases. In the databases, what you can store is that you can store the sequences, you can store images, you can store models, you can store patterns, you can store constraints, you can store any geometrical information, and so on. So this is a typical example of an amino acid sequence. Generally in the databases, in the biological databases, we store the single letter codes of the amino acids. This is a kind of a complicated stuff. So this is a kind of interrelationship between the databases. So if you see the sequence database, the sequences that are present in the sequence database, we can use those sequences to generate the phylogenetic tree, to generate the models, to generate the protein interaction networks, and so on. And this is not directly linked to biological data, but this is kind of when you are using the biological data to extract certain statistical features like disease network and so on. This is another very important biological data, the structures, the structural information of proteins. In the biological databases, as I told you earlier, that you can store the sequence as well as you can store the structures. So these are the kinds of the structures, mainly the structures of proteins or the nucleic acids or the protein nucleic acid complexes or protein nucleic acid ligand complexes, protein-protein complexes, protein ligand complexes. All these geometrical information, all these structure information are present in the biological databases. You will get to know more when we will do the hands-on session. This is not the biological data or very complicated biological data, mainly physics related, the biophysical data. Now, in order to perform various bioinformatics experiments, we need to know the specific patterns of the data that are present in the databases. Among the different patterns that are used or the different formats that are used in the databases, two of the most important ones are the EMBL and FASTA. because these two are the most widely used formats that are used in the biological databases, and these formats are easily readable by different bioinformatics tools. So we will focus mainly on these two. These are certain other file formats in the biological databases, and these are also useful, but they are used for some specific purposes only, like GCG. This is a specific file format which is used in the GCG package. That means there is a software package called GCG package which specifically uses this GCG formatted biological data. But interestingly, the GCG package can also use the biological data which are formatted in the form of FASTA. So for bioinformatics, basically these two formats are the most important ones. Any question up till this to the participants?
So there is a question in the chat box. What is microarray as shown in the previous figure?
Sorry, actually the voice is breaking. I can't hear you properly.
Sir, a question is in the chat box that what is microarray as shown in previous figure by Sopanmoy Coel.
Okay, let me see.
What is microarray as shown in previous figure?
In the chat box, I find only 3. Okay, what is microarray as shown in the previous figure? Okay, let me go back. Okay. This is not microarray. If you are talking about this figure, this is a vector and scalar field. This is kind of an information like the related to the magnetic behavior of a particular protein per se. So this is not microarray. And also, if I go back to here, probably you are talking about this or this, right? Microarray data means microarray data give us an idea about the expression profile of a protein. For example, some particular proteins are expressed more in one particular organelle. So microarray data give us an idea about which proteins are expressed in which particular organelle. Also, microarray data give us an idea about the protein interaction patterns. That means the patterns of interactions that are occurring in specific organelles. Sometimes a particular protein, for example, when a patient has cancer, there are certain biomarkers. Biomarkers are certain proteins. There are certain biomarker proteins which are expressed, highly expressed. The microarray data would enable us to identify which of the biomarker proteins is expressed in which type of cancer. So, okay, I think there are more questions.
Sir, we can go for the question in the end of the session for having question answer session.
Yes,
very good.
But probably I see 2 questions here. What is the importance of GCG and importance of GCG in bioinformatics? Sure, I'll give you the answer here. GCG is a particular pattern, of or particular format of data. And this particular pattern is used mainly in a package, in a software tool called GCG package. GCG package, whenever you want to solve a biological problem, with the help of the tool GCG, you have to feed or you have to use the data in a GCG formatted pattern so that the GCG package can identify the input data. But as I told you that GCG package can also recognize EMBL or FASTA format. Okay. I see. Are there any more questions? Okay. I don't think so.
Sir, we can take the questions which will come at the end of this session. So please, sir, proceed, sir.
Okay. And so
Hello.
talking
Yes, sir.
about,
Yes, sir. Sir,
uh,
please proceed, sir.
I believe there is
So there
another.
will be more questions coming. We can continue the talk, and later at the end of the session, we can have the question answer session, sir.
Okay. So I'm continuing then.
Yes, sir. Please. Sir.
Okay. So since so I was talking about the different types of file formats, the EMB, as I told you that the EMBL, SwissProt and the FASTA, these two are the most important file formats. So let us give you a brief idea about the other formats. ASN, it is called Abstract Syntax Notation Formats. It was used by NCBI. So, but now this ASN format is kind of replaced by FASTA. So now let me come to the EMBL SwissProt format. It starts with an ID. ID is nothing but it is a kind of identifier. It is kind of roll number with the help of which we can identify a particular data. And then it gives us an idea about what the type of data. It is a DNA containing 237 base pair. ID and the accession number are the same thing. But two different ways of expressing the same data. And then D line stands for, AC stands for accession number, D stands for description. RX stands for the cross reference. It is PubMed. This data, that means the experimental information or the background of this data is present in PubMed. FT stands for feature table. rRNA, that means 1 to 20 amino acids. I'm sorry, not 1 to 20 amino acids, 1 to 20 nucleic acids. They would produce the rRNA. And then it is the 18S ribosomal RNA. And then there is miscellaneous RNA from 21 to 205. It is that means the function is internal transcribed spacer. And SQ stands for sequence, having 237 base pairs. These are the percentages of the different bases. So this is a standard format. Many, or I should not say many, all of the computer tools, they can recognize this format, this file format. This is FASTA, a very, very important format. This format starts with this sign, and then there is a short description. And then from this line, we have the sequence. The sequence may be an amino acid sequence. The sequence may be a nucleic acid sequence. But the important thing is that it should start with this line. The computer program would identify this line to be the definition. So it would not, that means any line having this, this sign, that is being ignored by the computer program. That means the computer program will start processing the data from the next line. This is the FASTA format. And this is the GCG format. This GCG format is a kind of similar to the UniProt or the SwissProt format. There is The same thing, the description, the sequence, the ID, all those information are also are there in the GCG. But the difference between GCG and SwissProt is that SwissProt is more, that means SwissProt is universal. That means SwissProt format can be used for amino acids, can be used for nucleic acids, but GCG is mainly for nucleic acids. Then comes GenBank format. This is again, this format is being replaced by FASTA. This is Filip format, specifically used for phylogenetic analysis. But again, this format is not very important in the sense that nowadays we can use FASTA format in place of Filip format to perform phylogenetic analysis. And these are certain software tools. The details of the tools you will find here in these websites. These tools are basically to perform the basics of bioinformatics. And this is a very important paper appeared in nucleic acid research. And this paper provides a summary of the important databases that are present. And this is also for the basic, to perform the basic bioinformatics experimentations. And we will do this in the hands-on session. This is a screenshot of the literature database PubMed. And most of the biological databases are cross-referenced to this literature database. The literature database gives us an idea about the background information of a particular data. This is NCBI, the primary nucleic acid sequence database. And now we'll be moving on to GenBank, which is a part of this NCBI. GenBank is a primary sequence database. This why I'm calling this a primary sequence database, that is because whenever a new sequence is identified from any lab, that new sequence information need to be stored in GenBank. GenBank has collaborations with other databases. GenBank is basically a USA-based database, and there are the Asian database. The Asian counterpart of GenBank is DDBJ, and the European counterpart is the EMBL. So there are cross-talking between all these databases. A traditional GenBank record looks like this. The most important ones are these. This is the base pairs, and this is the type of nucleic acid, and this is the definition. This is the accession number. This is the source of the organism. This is the reference. That means PubMed. This is the PubMed. That means if you click here, that means this will directly take you to PubMed. So this is a kind of cross-reference. So see, certain things we need to know. The accession number. The accession number is very stable, and in all your like papers, accession number is accepted. This GI number is for the internal use of NCBI. See here, this is the accession number, which is universal. This GI number, though the accession number and the GI number point towards the same entry, but still in literature, in paper, we use the accession number. And the version, see, this is the version 0.1. What is the utility of this version? Version means, say, from one lab, the mRNA, this mRNA sequence was identified. Now what happens? After a few rounds of experimentation, more experimentations, there are certain modifications of the previously identified mRNA sequence. Those new modifications are incorporated in the database in the form of version. So 0.1 means this is the first incorporation. If there are more incorporations, then there will be 0.2, 0.3, and so on. Now, this is very vital information. This is the amino acid sequence from the gene, and the function of this particular protein, it is used in DNA mismatch repair. The gene length is this, the CDS is this, and another important thing is that the information regarding this protein only, this particular that means those information are stored in another database called SwissProt. So that is also a cross-referenced database. And this is the nucleotide sequence. The base count means number of A's and G's and C's and T's. This is another example, a shorter example. So now the question is, the information, the amino acid sequence information is already present in GenBank. Then why? What is the utility of having another database, SwissProt? That is because in GenBank, as I told you, this is a primary database. You have only the sequence information. But information, other information, that means the various annotations or the various features of this particular amino acid or the protein having this particular sets of amino acids, those features are not present in GenBank. They are present separately in SwissProt. So SwissProt is a kind of a secondary database which takes the primary information from GenBank and extracts the features from there and stores the feature. This is again the important aspect. This is the GenBank GI accession number, and this is the normal. Now, and the next one is the RefSeq, reference sequence. This is a very vital part of GenBank. See, the reference sequence is nothing but a particular information which is the most recent one. For example, from one lab, a particular amino acid sequence of a protein was identified. Then again, from another lab, the amino acid sequence of the same protein has been identified. Now what happens is, there is kind of controversy between the two because both the labs deposited the amino acid sequences of the same proteins in the databases. So which one to consider? Now, these people, they perform a separate experimentation and they try to identify the amino acid sequence separately using certain different methods. The newly identified sequence which matches with any of the existing sequences, that newly identified sequence is stored in the RefSeq, and that is considered to be the most reliable one. So the RepSec gives us an idea about the most reliable sequences. This is valid for amino acid, this is valid for protein, this is valid for nucleic acids, mRNAs, and so on and so forth. So you need to remember this NM. NM stands for, M stands for mRNA, NP stands for protein, NR stands for non-coding RNAs. This is XM, this is predicted mRNA. This is predicted protein. This is predicted non-coding RNAs. So from this, that means from the first two letters, we could identify, would be able to identify what type of information we are searching or what type of information are there in the database. So GenBank and RepSec, these two are highly interrelated, but the difference is that GenBank stores the primary information, all the information, but RepSec stores only those information which are valid. By valid, what I mean to say is that the sequences where no ambiguity is there. So this is a kind of wet lab technique by which the annotations, that means the RepSec sequences are observed. This is not actually bioinformatics. So the most important benefits of RepSec are that the RepSec is the validated one. The data are directly validated and non-redundant. That means there are no repetition. As I told you earlier that in GenBank, you may have multiple copies of the same data, but in RepSec, you have only one copy of the data. This is EMBL. This is DDBJ. This is a secondary amino acid sequence, Unigene, which is not very popular. This is also a secondary nucleic acid sequence database. This is for Saccharomyces cerevisiae only, very specific one. Now, these are the sequence related information. Now, from these sequence related information, what we can extract is that we can extract the different features. Mainly, the features are for for proteins, because nucleic acids generally have much variations, and their structures, definitely their structures are important, but not as important as the structures of proteins. So we will now move on to how to extract those features from the sequences of amino acids or proteins. But before I move on, let me see the questions. Which types of issues or problems related to biological data are dealt with in bioinformatics? Which type of issues? There are many types of problems, like extraction of specific sequence related information. For example, from an amino acid sequence, you can identify its possible cellular location, its possible structure, its possible function, and so on. Yes, CDS is the complementary DNA sequence. And how to predict the domain of protein motifs. This is done by several computational tools. Any other questions before moving on to the structural portion? Any other question before moving on to the structural portion?
Two question is that one question is that how to predict the domain of protein motifs.
Yes, I answered all these questions which are present in the question answer box.
Okay, okay.
Then there is one question from Pooja. Why there is CDS number is so long? Was it like why there is CDS? No, the CDS is the complementary DNA sequence. That means this portion is the gene from where that means which is converted to mRNA. The
No other
CDS
question.
is the coding portion. CDS basically represents the coding portion. The participants, can you hear my voice? Because I'm giving the answers. So shall I click on the answer live or type?
Answer live is fine.
Um,
Another question from Sushmita Biswas.
yes, the previous protein sequences which were identified before in other lab. No, GenBank stores all the information, whatever the sequences, whatever any lab can deposit the data in GenBank. But whether there is a conflict between the data or not, that is not verified. That verification is done in RepSec.
Okay, sir. Another from Pooja Samuel.
Yes, Pooja, yes, the coding sequence is basically the CDS is basically the coding portion. And the non-coding part is not there. So that means from the entire genome, if you know the CDS, then you can easily identify the non-coding part. Anything else? I have
Sushmita
asked
have
that
some.
lipstick is which one, the newly identified one or the RepSec is RepSec stores the information which which does not have any kind of ambiguity. It may be the older one, it may be the newer one. It is not necessary that the newer or the newly identified sequence is the correct one. If the previously identified sequence is the correct one, RepSec would take that into account.
Okay, no other question.
Okay.
Okay, sir. No other question.
No other question? Okay.
Yeah.
Okay. So let us now move on to the domains and the domain databases. So what is a domain? A domain is specifically an independent unit. It performs the most of the biological processes, and it has got several functional and evolutionary roles. That means basically a domain is kind of a part of a protein which helps the protein to exert its activity or functionality. Uh, the identification, how a domain is identified. There are several ways. Mostly, the domains are identified by sequence analysis and by searching the sequence databases. So the domains can be of these many different types. It is a single domain. It is an interconnected domain. This is a discontinuous domain, and this is interlaced domains. This is a single domain. This interconnected domain means this domain is linked to this domain by certain loops. This is discontinuous domain means this is one part of this domain, this is another part of the same domain. This part is linked to this portion, and this part is linked to this portion. And this interlaced domain means the individual domains, this is one domain, this is another domain. These individual domains have certain subdomains, and the linking between the subdomains are expressed as interlaced domains. Uh, there are certain things that we need to identify, like the false sequence relationship by local search. It may be that this is the domain information that we would like to know. But in the databases, we have these types of domain information which are similar to this, but not analogous or equivalent to this. So this is kind of false relationship. A domain size, it varies. A domain may have 30 amino acid residues to 200 or more than 200 amino acid residues. So that way, the domains may be classified as small domains, intermediate domains, and very large domains. So this is a kind of a graph, a relationship between sequence length and the expected the number of amino acid residues that are present in the domains. See, this way it varies. And also, as I mentioned in my previous slide, that large domains have multiple hydrophobic core regions. So this is an interrelationship between the sequence length and the number of hydrophobic amino acid residues in the domain. The domain characteristics. The domain characteristics, they vary between unicellular and multicellular organisms. In unicellular organisms, generally single domains are present, though it is not a thumb rule that the unicellular organisms or the lower organisms should have only one single domain protein. But in higher organisms, there are proteins having multiple domains, and these multiple domains have independent existence. That means the multiple domains, the functionalities of the domains are not dependent on each other. Domain fusion. It may so happen that the two domains may fuse together, may be combined together to come up with a new domain. And the new domain may have the functionalities, may perform the functionalities of both the domains. So this is a typical example of a domain fusion. This was an independent domain, this was an independent domain. These two domains were linked by this. So whenever there is a kind of fusion, a new domain will be formed having the functionalities of both this domain and this domain. See, what happens is this was a single domain. These were the portions, the active sites. Now, whenever another of this domain is fused with this, see, this becomes the active site. So certain new functions may arise, because here, see, the active site looks like this. Here, the active site looks a little bit different. Then comes domain swapping. It may so happen that the domain that is present at the left hand side that may come to the right side and so on. This is called domain swapping. So these are the different types of databases and that are domain databases. Main important domain databases are PFAM, Block, ProSite, and CAT and SCOP. These are the main important domain databases. These domain databases are based on the amino acid sequences, and these domain databases are based on the structures. So blocks database, that is a very old database or very basic database, which is being now replaced by ProSite database. ProSite is linked to SwissProt. This is not that important. Prince, it is also linked to SwissProt. It is dependent on certain fingerprints. That means certain motifs present in the particular database. So, see, let us look at here. These are the amino acid sequences of hemoglobin from different organisms. And this is a multiple sequence alignment of the amino acid sequences. From this multiple sequence alignment, we can come to a common portion that there is P at this position, there is Y at this position, there is V at this position, there is W at this position, there is T at this position, there is Q, R at this position. So this particular position is completely conserved. This is called a fingerprint. So all the hemoglobins should contain that particular fingerprint region. Prodome, then ProSite. ProSite database, it is a protein family database. This database gives us an idea, a complete idea about the different sequence features. For example, the most important one is the regular expression that I will show you in the next slide. See, this is a sequence alignment between, this is a protein X, this is a protein X from say organism Y, this is protein X from say organism Z, this is protein X from say organism A, this is protein X from say organism B. So we perform a sequence alignment. And from there, we could identify a regular expression like this. This AA stands for, at the first position, there should be either A or S. The second position is completely conserved, containing D. The third position contains either I or V or L. The fourth position contains G. Then there are 4 positions after G, 1, 2, 3, and 4. Those positions are variable. So X, any amino acid after G. Then 1, 2, 3, 4. Then comes this. It is expressed as PG. That means proline and glycine should not be there. Except proline and glycine, any other amino acid can come here. Then there is conserved C. Then there is either D or E. Then there is R. Then there is FY and FY2. That means first position either F or Y, second position either F or Y, and in the last position there is Q. This is called the regular expression. With the help of this regular expression, this is kind of a common sequence of all these. That means the protein X should have this type of common features or common alignments or the common pattern of amino acids. So These are certain other regular expressions that are present in ProCyte. Now comes PFAM, Protein Family Database. This is a very important database. In PFAM, what you can do is, if you can copy and paste a protein sequence in the query box, what you will observe is that you will find out the presence of the certain features in the sequence. That means to which family the protein belongs to. That will be identified by PFAM. So PFAM has such a huge utility. That means it gives you an idea about the presence of certain features, and on the basis of those features, the PFAM database classifies a protein to be belonging to one specific family. This is a kind of alignment, a multiple sequence alignment of PFAM. I think we have to move on to the hands-on session. So shall we move on to the hands-on session now?
You may, sir. You may,
Yes.
sir. According to this.
Huh?
You may, sir. You may do that.
Okay. Okay, let me see how many questions are there, and then we'll move on to that. Okay, in regular expression on the slide, which type of sequence alignment is done? It is multiple sequence alignment. Okay, this is in brief the different types of databases that we have. Let us move on to the hands-on session, and after the hands-on session, we will get to know more about the tidbits of all these databases, how to handle all these databases. So now I would like to request all the participants to open your internet. Okay, my screen is still visible, right, to all of you?
No, sir, the screen is not visible, sir.
Not visible.
Okay. Okay. Now is it visible?
Yes, it is coming, sir. So please
Okay.
choose the application, sir.
Okay. So see, I would request all the participants to kindly come to this website, to go to this website, ncbi.nlm.nih.gov. Once you open, you let me know that you have opened it. All of you opened it, right?
Yes, everyone is able to open it.
Okay, once you open it, then I will start demonstrating.
Sir, you may now do.
Okay. Okay. So now see, this is NCBI, National Center for Biotechnology Information, a primary sequence database. So if you come here, see, you will find all databases. So if you click here, you will find the different types of data that are being present here. So let us go to protein. Let us select protein and let us type this accession number. This is an accession number. Then press the search button. See, all of you opened it, right? All the participants?
Yes, sir.
Okay. See, this accession number is taking you to this. This is an operon. It contains these many different genes. It is coming from Pseudomonas salicylatoxidase. This, this data, this particular data are for a linear DNA having these many base pairs. This particular organism, Pseudominobacter salicylatoxidans, it contains these many different genes. This is the fourth version. Fourth version is the final version without any ambiguity. The details of the organism are present here. This is a bacteria. This belongs to proteobacteria and so on. The reference is here and this is the PubMed ID. So if you click on this, the PubMed, it will take you to this literature. The details, the experiment or the wet lab details to identify the operon are present here in this particular paper. It appeared from a lab, my previous lab in Bosch Institute. Here. This is the PubMed. So let us go back. So these are the names of the authors. Now comes the different features. From 1 to this is the genomic DNA. This is the strain of the organism. And this organism is present in Calcutta. So from 1st to 933, it codes for the gene SOX-G. The protein ID of SOX-G is this. So we will go to this. So let me first explain this. Then comes SOX-T. See, the SOX-G ends at 933. The SOX-T starts from 938. So between 933 to 938, these nucleotides are non-coding. This is the SOX-T. It ends in 2026. Then 2040 to 2415, it is SOX-R. then SOX S, then SOX V, SOX W, X, Y, Z, A, all these. Okay. Now, this is the nucleotide record. This is the single letter codes of the nucleotides. This is the entire nucleotide record. The single letter codes of the nucleotides. The single letter codes are always present in small, whereas the single letter codes of the amino acids are always present in capital. So that way we can discriminate between a nucleotide sequence and an amino acid sequence. So these are the amino acid sequences. See, as I told you earlier, that since this is a primary database, this does not contain too many annotations. For the annotations, we need to go to the Swissprot or UniProt. So these are the different genes and their corresponding proteins. You can easily identify. We'll go to UniProt once all of you get familiar with this. Once all of you get familiar with this, we'll go to the UniProt to have an idea about the annotations, different annotations. Let me know if you have any question here. Let me see in the question answer.
No, there is no new question.
Okay, so all of you were able to identify or able to get the knowledge, right?
Yes, sir. Yes, sir.
Um, now you can like search the NCBI using any accession number of your choice. Okay, so let us now move on to the UniProt. That means the database having the annotation. So let us click on here. Yes, see, this is UniProt data. This protein is called SOX-R protein. Here you will find many different information. This GEO stands for gene ontology. It gives you an idea about the molecular function. The possible molecular function is the DNA binding activity, transcription factor. This is a kind of a repetition. Pseudomonas salicylatoxidins, Pseudomonobacter salicylatoxidins. And this here, what you will find is that the family and domain information. The domain position is from 25 to 117. This is called helix turn helix type protein. See, there are cross-linking between the PFAM, the PRINs, and so on. The prosite. We'll see. This is the amino acid sequence. And this gives you this, this is another protein which has 90% sequence identity to SOX-R. This information is also present in EMBL. And a couple of things I need to show you is the prints and the PFAM. Let us go to PFAM. See, the PFAM is this. It contains, this is the result of PFAM. It contains the HTH domain, helix turn helix domain. So if you click on here, it will give you more details about it. This is helix turn helix is a structural motif of DNA binding protein. This is the structure, possible structure of a protein having helix turn helix DNA binding region. It binds to the DNA in the major groups. These are the major groups. This is the minor group. The function is mainly DNA recognition and transcription factor activities. So we will get more details of this helix turn helix protein from PFAM. So this is not only for helix turn helix, this is for any of the proteins. As I told you that PFAM would give you an idea about the possible family of a protein. So this protein belongs to the family of DNA binding proteins having helix turn helix motif. So this is called bacterial regulatory protein having R family. This is the heading. All the proteins having this particular signature, they are considered to be belonging to this family of proteins. Okay, then if we move on to the prosite. See, this is the entire amino acid sequence, the prosite. In the prosite, this is the domain 25 to 117. This portion is the HTHRR type. This is the DNA binding region. These amino acids bind to metal ions. So all these specific features are present. It contains a total of 121 amino acids. This is highlighted, this portion, 25 to 117. And this portion is also highlighted in green, DNA binding region. See, all these information were not present in NCBI. These are present in prosite, in UniProt. Let me know once you are done.
Yes, sir. From
Also
my side.
from your side. What about the others? Everybody? Shall I move on to the next then?
Yes, sir. Please, sir.
Yes, sir.
Um, See, these are very fundamental bioinformatics. So all of you should have idea about this. Now, this is what SOCs are. Then let us do another search for another protein, say SOX F. Again, let us go to UniProt. Here, this is SOX F. It contains a signal peptide. Its amino acid sequence is this. It contains a thioredoxin-like domain. But the problem is this is an uncharacterized protein. That means we do not have much evidence for this particular protein. So you will find this type of information in the databases as well, for which there are not much information there. For success type of protein, you can further work on this protein. For SOX R type of proteins, there are much less scope of performing experimentations, because most of the information are already present, are already known. But for SOX S type of proteins, you can work on it. See, you will have predicted, you are having uncharacterized proteins and something like that. The molecular mass of the protein is given here.
Sir,
Yes, yes, go ahead.
please, sir.
Yes, what's your question?
Sir, I'm just telling you, sir. Please proceed, sir. Sir, everybody is listening, sir.
Okay. Then let us take a little bit bigger protein, because all these proteins were smaller. Let us take this one. This is SOX A. It is a little bit bigger. See, here, since it is a bigger protein, there are much scope of annotation. It is a C-type diheme cytochrome. It performs this type of catalytic reactions. It contains Fe3 plus as cofactor. So Fe3 plus is converted to Fe2 plus. It reacts with thiosulfate with the help of this cysteine residue. So there is direct evidence of the biochemical reaction. Here you have the information regarding the binding sites. See, at this position there is heme. This position iron binds. At this position the substrate thiosulfate binds, and so on. Its molecular function, you have many molecular functions. It is involved in metal ion binding. It has got relation, that means it has its activity in oxidation reduction reactions, and so on. In the biological process, it is involved in sulfur oxidation. The subcellular location, it is present in cytochrome and in periplasm. This is the signal peptide. This particular domain is present, cytochrome C domain. This is the amino acid sequence. This is a similar protein. So the larger the protein, the more information you will have. The smaller the protein, the less information you will have. Okay, up till this is clear? All of
Yes,
you,
sir.
these are very basic things that you have to be familiar with if you want to do bioinformatics.
Yes, sir. These are very clear.
Okay.
Yes, sir.
Okay. So, is there any questions? I believe someone asked me a question. Okay. So now from here we can go to FASTA. And this is the FASTA sequence. This is the FASTA sequence of the nucleotide. See, in the FASTA sequence, as I told you earlier, that it should start with this sign. So this is the FASTA sequence of the nucleotide. You can use this FASTA sequence to any of the, that means this FASTA sequence can be taken as an input to any of the bioinformatics tools. Also, if you want, you can click here in the protein ID.
Unshaman.
Huh. Hello.
Unshaman, can
Huh.
you listen to me? হ্যালো,
Hello. হ্যাঁ বলো।
হ্যাঁ একটুখানি তুমি কি বলবে আমরা যখন কাজ করি তখন
হ্যাঁ।
এতগুলো সিকোয়েন্সের মধ্যে ফাস্টা ফরম্যাটটা ম্যাক্সিমামই ইউজ করা হয়, মানে
হ্যাঁ।
হোয়াট ইজ দ্য রিলেভেন্স অফ, বা আমাদের মানে কি বলবো, হোয়াট ইজ দ্য ইউটিলিটি অফ ইউজিং দিস ফাস্টা সিকোয়েন্সেস?
হ্যাঁ,
মানে এইটা যে সোজা ফরম্যাটটা বা এই এই যে জিনিসগুলো আর কি, এটা যদি একটু বলে নাও তাহলে বোধহয় ভালো হয়।
Sure. But the main importance of the FASTA sequence is that it is very easy to read. Any computer program, if you just go to the FASTA sequence, what you will find is that it's a very simple sequence. The first line gives you the entire description. This is the protein ID. This is the name of the protein. This is the organism from which this protein has been extracted. And from the second line, you will get the amino acid sequence. So if you are using this FASTA sequence, only this much of information is necessary to make you understand or to give you an idea about the entire protein. And all the tools, all the available tools and that are used in bioinformatics, all those tools can identify this or can easily relate or can easily read this sequence format. Because in the FASTA format, see, whenever there is this sign, the tool, the bioinformatics tool that you are using, it would automatically consider the first line to be a kind of description line. So it would ignore that line and it would start processing the sequence from the second line. So like you did not have to make the tool understand that, okay, you should go to this line to carry out the experimentation or to process the sequence. That is the utility. This is the FASTA sequence of this SOX-R protein. And see here also, you have the single letter codes of the amino acids. Now, everybody get my point?
Yes, sir.
Uh,
Yes, sir, I got it.
so let us, from sequence, let us move on to the structure. So let me see if you have any question regarding these sequences. Okay, I see a couple of questions. Okay, one question I think I see. What does the numbers beside the origin denote? Okay, this is from Shamurjit Malik. Okay, the numbers beside the origin. Let us go back. This number 1 means this is the first nucleotide. This is the first nucleotide. This is the 60th nucleotide. The second line starts with 61st nucleotide. It ends in 120th nucleotide. The third line starts with 121st nucleotide, and so on. These are the significances of the numbers. Okay, Sharojit, Shamurjit. Achha. Mitrabroto, sir, how to know a specific reference number to put in NCBI? A specific reference number, I think you'd like to know the specific gene or a specific coding region, so that you can identify from here. See, say you would like to know the SOX-D. It starts from the position 9735 to 10874. So 9735, if I go back here, 97, here it is 999897. It starts from 9721 and ends in 30. So 31, 32, 33, 34, 35 here. This portion from this A, it is 9735. So that way you can identify the specific portion. Okay, Mitrabroto. Any other question?
No, sir.
Any other question from this sequence portion? Okay, in chat line also. Um, okay, so any other question? Okay, Shushmita, how could we annotate a genome when dot? This question is not very clear to me. How could we annotate a genome where .fna files? I'm not talking about .fna files or .faa files. Those are something else. Those are not related to NCBI. In order to process those files, you have to have some specific software. Okay. So let us now move on to protein structural database, and that is PDB, a very vital, very important database. Everybody, this. Everybody should come to this. Once you get it, I will start. This is PDB.
Yes, sir.
So PDB. Here also, we can search the PDB using a PDB ID. That is, let me show you one PDB ID. We'll search with that, and then we will do a generic search. So this is one PDB ID, one SMT. One SMT. See, in this line, it is written as this a description. It is called SMTB repressor, and the organism is this. This is a metalloregulatory protein. It is obtained by X-ray diffraction method with a resolution of this. This is the literature. You can search PubMed using this ID. Oh shit, there is a problem. Don't know whether it is my network or yeah, I think it's coming. Yes. This is the paper which would give you the wet lab details of the identification of this particular protein. the extracrystallographic details. And you will find the abstract here as well. Here you have a certain brief description. This is a transcriptional repressor protein. It contains 2 chains having a sequence, amino acid sequence of this. These are the details of the experimental methods. Here is the structure of the protein. It contains the helical regions, the sheet regions, and the loop regions. Okay, all of you got it?
Yes, sir.
So now let us move on to annotations. See, in the annotations, what we observe is that there are the domains by scope and cat. I told you while I was talking about the structural databases or the structural domain databases, that scope and cat are the 2 of the most important structural classification methodologies. So here it is an all alpha protein, the class to which the protein belongs. It contains a DNA or RNA binding domain. It contains a winged helix DNA binding domain. Winged helix DNA binding domain means the helices are linked to an antiparallel beta shape. This is RSAR-like transcriptional regulator. This is for chain A, this is for chain B. The similar information are present in both SCOP and CAT, but expressed in a little bit different way. Here, it is expressed as class, fold, superfamily, family, domain. Here it is expressed as class, architecture, topology, and homology. But they would represent the same type of data, same type of information. The basic idea is it is an alpha helical protein. It belongs to RSAR-like transcriptional regulators, and it binds to DNA. Then family annotation. This is a regulatory protein. It is present in bacteria, and a DNA binding helix turn helix motif is there. Then GO. These are the molecular functions. It binds to DNA. It binds, it has its tendency to bind to metal ions, to bind to heterocyclic compounds, to act as transcriptional regulators. So these are the different molecular functions. So these are the annotations that are coming directly from the structure. The difference between the annotations of sequence data and the annotations of structural data are that from structural data, you will get more direct annotations. From sequence data, the annotations are mainly probabilistic. The annotations are based on the other related sequences that are present in databases. But from structural data, you can have direct access to the structures. And by analyzing the structures, you could specifically identify the functions and the other related information. I think there is one question again. Probably this question is not directed to me. I can't give you the answer of this question. Okay. Shall I proceed?
Yes, sir. Please.
Um, but I'd request you to kindly let me know so that I can proceed. Otherwise, I can't understand that whether you are getting the points or not. Okay, so let's move on to the sequence. See, this is the sequence information. From this sequence information, if you can see it clearly that these are the regions. This amino acid to this amino acid, this portion would represent a helical region. This portion helical. Then comes the beta strands. These portions would represent the beta strands. These portions, the metal ion binding portions. So the DNA binding region. So all these, all the information that we found in the annotation sections, those are again presented here. But here you have direct access to the sequence. And you are kind of comparing the sequence with the structure in a direct manner.
Yes, sir.
Yeah. Okay. Then I will show you. These are kind of an overview, but how that means when you download this particular PDB file, how it looks, that I'm going to show you now. PDB format. See, it looks like this when you download it. It starts with the same annotations. All these annotations are already present in the structure display portion. but the most important part is here. From here, the most important part starts. This is atom record. This is the first atom. The first atom is a nitrogen. It is from glutamic acid. This is the chain. This is the 24th amino acid. And these are the X, Y, and Z coordinates.
Okay, sir.
X, Y, and Z coordinate. This is called the B factor, and this is the probability factor. B factor means see the atoms, I'm not an a strict stenographer, but just to give you a rough idea about the process, is that uh, whenever you try to identify a structure, you are having X-rays, and the X-rays are interacting with the electrons, but the electrons are always moving. So it is really difficult to identify the position of an electron. So the electron of this nitrogen atom, um, they're moving, and so on an average, uh, around like 45% of the time, you are getting the coordinates at these positions for this nitrogen atom. This is Ca stands for C alpha. The same things are here. And these represent that we are sure that this is the nitrogen atom. This is carbon, this is oxygen, this is C beta, this is C gamma, this is C delta. These are the side chains. This is the main chain, N, C alpha, C, and O. These are the side chains. So the 24th amino acid, glutamic acid, is completed here. Then comes 25th, starts with nitrogen, leucine. N, C alpha, C, O, NCl. Then C and side chains. It ends here. Then 26th, glutamine. 27th,
Okay,
alanine.
sir.
Yes? Okay, question?
No, sir. No questions.
There is one question, Nabonita Bishwash. How to find the active site of a protein for docking? Active sites are present in the PDB. Like in this particular case, the DNA binding region or the metal ion binding region, whichever region you want to target, the amino acid sequences are directly there. So you can relate the amino acid sequence with the domain information. So this is the way. The most important part of a PDB is this portion. This is the here the PDB is different from others. You get the structural details here. Tar means the first chain ends here. Again, the second chain starts here, and the B. B stands for the B chain. This is chain A, this is chain B. So it ends here. So that way, this is how a PDB file looks like. You can ignore this portion. This header information are already there in the structure summary. You can directly go here, go to this part. So then come to the 3D view. This is, I think it is it will be a little bit difficult because the 3D view requires certain other tools. I don't think I have that one installed here, but if in your laptop, if you have those Java applications, you will be able to find out the structure. You'll be able to get the structure here. It is present in a very nice way.
Yes, sir.
So probably because in my case, I don't have the Java application installed, so I can't show you here, but in your laptops, you might have, so you might see the structure. So 3D view is a special feature of PDB. This one will be in front of you, and you'll be able to interact with it. This is one PDB using an accession number or a PDB ID. Now, let us search the PDB database in a generic way. Generic way means not using any accession number. So let us start like this. Source which is very hot these days. See, for SARS-CoV-2, these many structures are there. And not only X-ray diffraction, there is electron microscopic structures are also there. And this is the RNA genome, the structure of the RNA genome, not the protein. So though the name is a protein data bank, it stores information about the nucleic acids as well. So let us see, this is a very recent one, appeared in 19th of August. This is the main protease, and see, let us see, this is the protein. It has not yet been published. Chain A, it contains 306 amino acid residues. Now, what I would like to show you is that this protein has this non-protein part. This is a ligand, which is bound to the protein. This is dimethyl sulfoxide. This is kind of a solvent, but this is the most important one. This is the ligand. And the ligand is a peptide-like ligand, and it is an enzyme inhibitor. You can click on that. See, this is the structure of the ligand. It binds to the protein. And if you click on the ligand interactions, here also we need Java tool or Java app to see the ligand interaction. But if you have the Java app in your laptop, you'll be able to find out which amino acids are directly interacting with the ligand.
Yes, sir.
You can check it in your laptop if you have the Java application. Okay, so what new information? Let us move on to the annotations. See here, again, the protein family information are there, the details of the family information, the molecular function, the biological process, and the cellular component. In the previous case, there were no inhibitors or no non-protein part, but here you have the non-protein part, that is the inhibitor. This.
Yes, sir.
Let us go to the experiment. Let me show you. Um, this is the experimental, the crystallization methods, what pH and temperature are used, what is the solvent, and the other properties. Not so much of importance to the bioinformaticians. Then comes the sequence. See, here again, the helical regions, the beta strands, the covalent bonds. That means how the different amino acids are linked together. Here, the covalent bond means maybe the disulfide linkages or the linkage between if the ligand bonds binds to the protein through a covalent linkage. But generally, the ligand does not bind to the protein through covalent linkages. Generally, the covalent bond means it is a disulfide bond. These are the active site regions. These are the different domains of the protein.
Okay.
So, so from PDB, what we can find out is that we can easily identify, very easily identify the different structural features, which are that means this is the most reliable one. Why I'm telling this is the most reliable one? That is because we are getting the information directly from the structure, and these are not dependent on any speculation. Now, let us see. Okay, now let us see the structure of this nucleic acid. See, this is structure of the nucleic acid. The length is 88. Electron microscopy was used here. Let us go to annotations. See, for nucleic acids, you generally don't find any annotation. Why? Because nucleic acids generally do not take like and do not perform any types of catalytic reactions. This is not the right way to tell you that nucleic acids do not take part in any of the catalytic reactions, because nucleic acids are involved in transcription and then the translation. But the main mediators of these reactions are the enzymes or the proteins. So the proteins have much more variations, and the variations in proteins occur due to the like the presence of 20 different types of amino acids, and they can be present in any of the combinations. But for nucleic acids, only 4 different types of nucleotides are available, so the number of variations are much less.
Okay, sir. Sir, is there FemSit stimulation does any kind of annotation changes in this kind of proteins?
No, actually, it is for the nucleic acid. That means this is not the protein. So they are these people, they identified the structure of the nucleic acid only of the genome, the structure of the genome. So not the structure of the protein.
Okay, sir. But FemSit mutations or simulations can be done in nucleic acids too, na, sir? Instead of proteins.
It
So
does not.
is it relevant these FemSit simulations are capable to done some changes?
The
Hello.
changes
Sorry, sir, I
are
can't hear you.
very important because the changes are not reflected in any of the biological activities, because the biological activities are mediated by proteins.
Okay, sir. Thank you, sir.
Let me show you the PDB format. Here also the same things are here, and here instead of amino acids you will find the nucleic acids. This is G of chain A, this is U, this, since this is RNA, so this is U, chain A, and so on. And these are the coordinates. The same thing as a protein. And here also you can see the FASTA sequence. See, this is the FASTA sequence in the same manner.
Yes, sir.
Uh, okay, let me see if there is any question. Okay. Okay. So these are the structural features, structural information that are available in see in chat line. Okay, so now let me show you another structured database, which is not PDB, but that is called NDB. NDB is a nucleotide database. So, but it is not as popular as PDB. So, I would tell you to go to Google and type this nucleic acid database. It is not very popular as PDB, but if someone is very much interested in nucleic acids, in finding the structure of nucleic acids, both RNA and DNA, you can search here. Here, you can search for DNA, you can search for RNA, you can search for, you can perform any of the advanced search like DNA protein complexes. So let us do a search here in the advanced search portion. No, it will be a little bit difficult. So let us type here. DNA. See, here you will find all these crystal structures. Now, if you click here, you will get the protein DNA complexes, the structures of protein DNA complexes. And another very important aspect is the drug DNA complexes. Since drugs are mainly targeted to proteins, but here there are certain drugs. See, these are the very new structures. Certain drugs, they are directly targeted to nucleic acids. For example, here, if you click here, see, this is a polythymine DNA. It does not contain any protein sequence. It contains only the DNA. It is obtained by X-ray crystallography. See, this is the sequence. This portion is very important. You can search RNA in a similar manner. All these are RNA.
So,
For any specific type of RNA, like ribozyme, you can search here as well. The ribozymes, the catalytic RNAs. Or the protein RNA complexes. All these are there, RNA binding proteins. Drug RNA complexes, very vital one. See, no results. So this is another structure specifically meant for nucleic acids only. But as I told you that this is not very popular, as see, all these are also present in PDB. See, the corresponding PDB IDs are there. Corresponding PDB IDs are also there. But if you want to, this is specific for nucleic acids. So if you in PDB, if you want to search, say, let me show you. You can search it like this, DNA protein complexes. You will find so many different results. There are 25, so many different results. So 6722, these many types are there. But like specifically, you can use NDB to identify only the nucleic acids. But as I told you, PDB is more popular. You can search it in any way. Okay, so let me see if there is any other question.
So,
Yes.
sir, I have one question. Sir,
Yes.
in that NDB, at the first very page you have shown, there are some PAM sites mentioning there
Mhm.
with their sequences, which we didn't get in PDBs. So in
Yes.
NDBs, we can get those spam sites, and after getting those spam sites, we can design primer against them in
Absolutely.
modern technologies like RNA silencing and CRISPR.
Absolutely. You can use that. As I told you earlier,
So
that
every PAM sites are
yes, PDB is mainly focused on proteins. It contains nucleotide information. It contains protein nucleic acid complexes, but not all the details are there. The nucleotide related details are not there in PDB.
yes, sir. So this thing is very much helpful for primer designing also.
Yes, you can use
Thank you.
that.
Thank you, sir.
So let us then go to the slides again. Uh, See, I stopped here just to show you how that means these databases function. So this is a multiple sequence alignment of PFAM, and this is interpro. So I'm not talking about all this. Yeah, cat. Cat. The name implies that class C stands for class, A stands for architecture, T stands for topology or fold, H stands for homology or superfamily. So as I just showed you that cat and SCOP, these are the structural classification of proteins. So it covers these alpha, beta, alpha slash beta proteins. Alpha, beta, that means the protein which is majorly alpha helical. Beta means which is majorly beta sheet. And alpha slash beta means it contains both alpha helical and beta sheet regions. Uh, this is also not so important. SCOP. Because SCOP is another way of classification of proteins. It classifies the proteins as all alpha, all beta, alpha plus beta, and alpha slash beta. So let me show you what are all these. All alpha means the protein should contain mainly alpha helical regions. It may contain beta sheets, but the extent or the percentage of beta sheets should be less than like 15% of the entire amino acid sequence. Similarly for all beta. Alpha plus beta means this is a protein which contains both alpha helical and beta sheet regions, but the alpha helical and beta sheet regions remain separate. And alpha slash beta means this is or this protein contains both alpha helical and beta sheet regions, but the alpha helical and beta sheet regions are intertwined. That means they are linked together, they are joined together. So, so CAT and SCOP and basically give us an idea about the relative distribution of the secondary structures in proteins. But they are very vital because from all these information, we can identify the possible function and cellular location of the protein. So that is why these CAT and SCOP information are also present in PDB.
Unshaman,
Ah,
may I say one thing?
huh?
May I say one thing? One addition.
Huh?
As you were saying alpha plus beta and alpha slash beta, you can say that alpha slash beta, as already you have mentioned, that is a Rossmann fold. There are
Yes.
the, if you are just mentioning this thing, there are particular folds which are the super secondary structures. I think that will be little helpful for them also.
Yes, absolutely. So uh, this is a kind of basically the structural distribution, the secondary structural distribution of the protein, that means how the secondary structures are present in the protein, and see, the classifications are not very rigid, because in there is kind the difference between all these classes. All these classes are kind of uh, that means we cannot generally so easily discriminate between the different classes so easily, because the relationship, there is a kind of common interrelationship between all these structures. Um, excuse me. Uh, like for example, mainly alpha plus beta, mainly parallel beta sheets are there. Parallel beta sheets. I think all of you have an idea that what parallel beta sheets are there, what parallel beta sheets are, and a kind of alpha slash beta, there is an intermixing of these types of beta sheets. But as Professor Banerjee told you that these are fold specific, like the Rossmann fold or the different protein folds that are present. So these classifications are based on specific types of protein foldings. And the protein foldings are like guided by the intermolecular forces of attraction among the amino acid residues. Okay, so if there is any other question, any more questions, I'd like to address all those questions. Any questions? Any question? Um,
Sir, already all the questions answers have been given, sir.
yes, in the chat.
Yes, sir. It is very informative. It is very good.
The questions, I answered all these questions that I find in the question answer box.
Yes, sir. In the chat box, there are only one question that has been already answered. We request to the participants, if they have any questions, they can post it in the chat box or in the question answer section.
Or you can unmute your microphone and ask me.
Yes, sir. By raising hand.
There is one question I find, database for DNA origami structures. You can search it in NDB.
Somin, sir.
Yes.
Yes,
Sir, almost I
actually,
think we think that
I
the
think
questions have been answered, sir.
so. All, Angshuman sir,
Yes.
already all questions are already answered. So thank you very much. Thank you very much, Dr. Bakshi, for your restless, wonderful session. It has been a very useful session to all the students and researchers, I believe. Students have learned about what exactly are databases and how they would see the biological data through these databases. But the most important aspect is the central dogma, which is something all bioinformatician or students taking up this subject should understand. Dr. Bakshi has covered very deeply in different types of data like nucleotide, amino acid, DNA, RNA, etc. Then classification all these databases and where these databases are available, what formats of biological data are to be used when and one uses these databases was explained very well. So thank you very much. It has been very interactive session through the question answers put forward by our students. Thank you very much, Dr. Bakshi, for your wonderful session.
Thank you all.
Tomorrow it
Uh,
will be
now,
continued.
Professor Banerjee to share his valuable views on today's session before the audience.
No, this is very interesting, and this is, I think this is very helpful for the who are and also who are not in the bioinformatics line. Basically, Dr. Bakshi has mentioned some of the idea. This is a pseudoscience like this thing, but this is not actually pseudoscience. That will help. This bioinformatics basically helps you through the computer programming and all other things, that is, which is not really viable or which is not possible to do in the wet lab experiment. So this one thing we have to keep it in mind is this bioinformatics is also an experiment which we are doing in not in vitro, but we are doing not in vivo, but we are using that in silico. And this is the very important thing. And actually what he has told you, this is like bricks. You can see the palaces, you can see the houses, you can see the hearts, but everywhere there is a bricks at there. But the thing is how you arrange all these things, depending on that, whether you will get the Taj Mahal or whether you are getting the simple things. So simple, this protein are these things, these are nothing but this is the basically the hierarchy of the structures. Actually I have interrupted from Dr. Bakshi, sorry for that. So that is one thing is basically very interesting, how this small thing, basically you can consider the few words of Shakespeare, little grains of sand and a little a few drops of water make the mighty ocean and the pleasant land. So the proteins, the big thing is there, but the small things are amino acids. The DNA is big, but the small thing is the four nucleotide bases. So how they are arranged, if you do not know this basic thing, it will be very difficult to know. So once again, thank you so much, Dr. Bakshi. And I hope next day, tomorrow, we will continue with this, where we will see the more modern trends and how to apply this thing to the in the research part. So once again, from my side, from the side of the university, we extend our sincere gratitude and the sincere thanks to you for coming here.
Okay,
Thank
thank
Dr.
you,
Putin.
thank you.
thank
Now,
you.
Kumar, is there any announcement for the participant?
Yes, sir. Sir, we have posted the registration link for tomorrow session, and
Yeah,
the quiz and feedback link for today, sir.
yeah.
And that is also at 10.
Shall I share it right now?
Please wait, sir. Sir, let Somin sir give the permission. Then only.
Okay.
So tomorrow also at 10 a.m.
Yes, sir.
Yeah, yeah.
Tomorrow at 10 a.m. Yes, sir.
So the participant, you please fill up the feedback form with some multiple choice question for the limited outcome of this session. And this is mandatory for all the participants. Hope you will enjoy the today's session and the session may be formally closed. We will meet tomorrow at 10 a.m. positively.
So now share the
Thank you
links.
very much. So see you all tomorrow. Thank you.
Okay, okay. Thank you. Thank you. Thank you to all. Thank you, Professor Bakshi.
So now share. Can I share the link now?
Yeah, yeah.
Sir, one more announcement is there, sir. For certificates, certificates will be given tomorrow, sir. After
Yeah.
tomorrow's session.
Prem, is it visible?
Yes, yes.
Yes.
It is requested to all participants to kindly take note of the registration link for tomorrow session, day 2, and the quiz for today and the feedback link. And please do register for tomorrow's program using this link. We have posted the links in the chat box also, and it is visible in the screen also. Participants can take the screenshot of these links, and the certificates will be provided tomorrow after the completion of the 2-day workshop.
Okay. It's clear to all.
Yes.
And also given here the YouTube link. You can watch today's session in the YouTube link. The YouTube link is given here.
Yes, sir. কাকু আমি একটা ওয়েবিনারে আছি, আমি তোমাকে একটু পরে কল ব্যাক করি। হ্যাঁ, ঠিক আছে।
The registration link for day 2 workshop has been displayed in the screen and has been posted in the chat box. So please, it is requested to all participants, those who have participated for day 1, please do register for day 2. After completing the day 2, we will be providing the certificates.
And you must fill up the feedback form with quiz. Okay, I think you must take a screenshot of this page for registration purpose and other. Okay, thank you to all participants. We'll meet 10 a.m. tomorrow positively.
Shall I stop share now?
Yeah.
Sir, with your permission, can we close the session, sir?
Yeah. You may close.
So the next day 2 session will be tomorrow at 10 a.m. Thank you all. Thank you, sir.
Thank you. Thank you, Prem Kumar.
Thank you, sir.